Bacterial Transformation, the final lab presentation for Biology 110 from Cheyenne. In this course, we really had a wonderful time in the lab. We practiced using micropipettes, did experiments with the lactase enzyme, observed the mitosis of garlic cells, and did the nitrogen test. I even brought home 4 garlics to grow on my window ledge. But here, I choose to do the presentation about bacterial transformation since our professor was especially handsome on that day. Just a joke. I like this experiment anyway. So let's take a look at some basic concepts about bacterial transformation. As we know, bacteria are prokaryotes, which means they do not have nucleus envelope. Instead, they contain DNA in an area called the nucleoid. Transformation is the process by which exogenous DNA is introduced into bacteria and then expressed as functional proteins. A plasmid is a small, circular, double-stranded DNA sequence found in bacteria that codes characteristic proteins. In recombinant DNA research, the multiple cloning site, MCS, as well as the origin of replication are needed in the plasmid. And of course, the foreign DNA can be either a sequence to be embedded in the original one or as an isolated plasmid existing in the cell. So, which cells can pick up foreign DNAs? Well, the competent cells. There are 2 primary methods of artificial transformation in the lab. The first one is chemical transformation for chemically competent cells, and the second is electroporation for electrocompetent cells. Now, let's step into the biology lab. Bacterial transformation requires several steps. The first step is to prepare competent cells. Since the cell walls and membranes are negatively charged, we use divalent calcium ions to increase the permeability of the cell walls and membranes. During transformation, the heat shock results in the formation of adhesion zone, enabling the cells to take up plasmids. After recovering, we can deliver them to the plates. But how do you distinguish transformants from normal cells? Well, we can use selective markers or integrate other genes in the plasmid. Specifically, the selective marker here is the ampicillin-resistant gene. As you can see, ampicillin is an antibiotic that inhibits the growth of E. coli. Here, we introduce the plasmid called PUC19, which contains the ampicillin-resistant gene as well as the LAC-Z gene. LAC-Z gene allows the blue color colonies to be exhibited. The relationship is a bit complicated, so I drew a graph here and I believe you smart guys can figure it out by a simple screenshot. The heat shock might be one of the most important steps in the protocol. After adding the plasmids to the cell solution, we put them on ice for 20 minutes, and then deliver them to heat at 45 Celsius for 45 seconds. After that, we add some growth factors, and they're ready for further incubation. Here comes the exciting 45 seconds when we have to be super careful to take them out quickly. Here comes the recovery process. In the incubator, the tubes are kept at 37 degrees Celsius.
So now I have done the microcentrifuge and I'm going to remove the supernatant.
The microcentrifuging is important because it can increase the concentration of cells. Here I'm going to plate the cell solution onto the plates. glass beads are used to make sure that the cell solution is evenly distributed. Here comes the exciting results. As you can see, one plate exhibits blue colonies, while two other plates exhibit white colonies, which means our experiment has succeeded. What about its real-life applications? Well, bacterial transformation can be used for DNA cloning as well as the synthesis of certain proteins. For instance, transformants are used to produce insulin, which can then be utilized to treat diabetes. Nowadays, bacterial transformation has played a crucial role in medicines and pharmaceuticals. That's the end of my presentation. My ass. I prepared a bonus for my garlics.
Look, my baby garlics are thriving. Ooh. Nice.
3 weeks after the initial experiment. That's all for the presentation, and thanks for watching. I really enjoyed the lab experiences in Biology 110